Welcome to the third webinar in the Psychedelics and Mental Health series hosted by the Center for the Neuroscience of Psychedelics at Mass General Hospital. Today's program features Dr. Stephen Haggarty, Scientific Director of Chemical Neurobiology for the Center. Dr. Haggarty is the Stuart and Suzanne Steele MGH Research Scholar with appointments at Harvard Medical School, the Broad Institute, and MIT. He'll take us on a deep dive into the power of MGH's cutting-edge chemical neurobiology and how he plans to bring those technologies to bear on a library of Harvard's unique collection of psychoactive plants. This work will help to better understand the ways that novel psychedelic compounds can facilitate lasting changes in mental health, also potentially neurodegenerative diseases, including Parkinson's and ALS, and possibly Alzheimer's, as well as overall well-being. Dr. Haggarty's presentation links the pioneering work from the history of psychedelics with state-of-the-art science and technology to help define the future of psychedelics and brain health. For those who are new to the series, I suggest you also listen to the earlier Salon recordings for an overview, as well as a better understanding of the amazing neuroimaging capabilities at MGH. Today, you'll get a behind-the-scenes look at one of the key pillars of the center being brought to life at MGH, Harvard Medical School's primary teaching hospital and the hub of the Boston biomedical ecosystem. I'm Dick Simon, chair of the center's advisory council. I spent all of my time doing what I can to advance psychedelic therapies for treating a wide range of mental health issues. I'm motivated both by the astounding data supporting the efficacy of psychedelic therapy and from having experienced the seemingly intractable nature of mental health issues in my own family. I'm excited about this center with its unparalleled human and technological resources, as well as its integrative approach of psychiatry, cognitive neuroscience, neuroimaging, and chemical neurobiology, the subject of today's salon. As a reminder, at any point you can type questions at the Q&A button and we'll address them during the question and answer. Now I'm happy to introduce my good friend, Dr. Jerrold Rosenbaum, founding director of the MGH Center for the Neuroscience of Psychedelics. Dr. Rosenbaum is psychiatrist and chief emeritus for MGH's psychiatry department, which he stewarded as the top-ranked psychiatry department in the United States for 20 years, and a professor of psychiatry at Harvard Medical School. He brings 47 years experience as one of the preeminent psychiatrists and psychopharmacology experts in the world. Jerry.
I want to thank Dick for his being an extraordinary patron and advocate for the field and for our center. And in a moment, it's going to be my distinct pleasure to introduce my extraordinary colleague, Steve Haggarty, who is the scientific director for neurobiology of the Center for the Neuroscience of Psychedelics. At MGH, we were inspired by the idea that the class of agents known as psychedelics can offer a window on how to move the brain to change in a way that can address many of the most anguishing forms of human suffering. So we are mobilizing the extraordinary resources of the Mass General Research Institute and Mass General Neuroscience at the largest hospital-based biomedical resource in the US, with well north of $1 billion of annual funded science, to go through that window on the brain that these molecules can open. And once inside, by understanding the neuroscience of these effects, we will dramatically advance the efficacy and precision of therapeutics for brain disease. As many of you have heard before, the center has multiple dimensions through which it will take on this challenge, and today's webinar will focus on one aspect of the work of our Director of Neurobiology, Steve Haggarty, who will address the topic of ethnobotany and psychedelic discovery. Again, we will take a number of questions. We'll leave time at the end for questions, so please take note of the Q&A function at the bottom of your screens. And now it's my pleasure to introduce my colleague and friend, Steve Haggarty.
I'm just delighted to be here this afternoon and have the chance to speak to you all about the work, as Dr. Rosenbaum just introduced to you, that we're engaged in within the Center for the Neuroscience of Psychedelics. This is an area, I think, of long-standing interest for myself in terms of the potential contributions that the field of chemical neurobiology can play in this really exciting area of translational neuroscience and therapeutic applications. When thinking about how we can bring translational neuroscience into the center, we conceived of a vision here in which we would be able to build a collaborative network of research investigators who are focused on advancing our fundamental knowledge of the molecular, cellular, and network level mechanisms of neuroplasticity that, as Jerry just described to you, psychedelics can modulate. Our goal here is really to advance then the ability to use precision medicine concepts in the context of psychedelic therapy, which we might summarize as considering the right drug for the right patient at the right time. Now, before I get into the science of what I want to share with you today, I want each of you to just think for a moment of whether you yourself have consumed a psychoactive plant today. This exercise is usually a little more fun in person to see he or she that will raise their hand bravely to share with us their experience. So think for a moment if you did. And I'll begin answering myself that question and admitting that this morning, in fact, this afternoon as well, I consumed some coffee, extract then from a bean of a plant known as coffee arabica. Those of you joining us on the webinar here from the United Kingdom a little later in the afternoon, or perhaps you're in India or China, we might have consumed a black tea then, Camellia sinensis, then, a widely known and widely consumed psychoactive plant. Those of you that are joining us perhaps from South America, in the countries of Argentina, Uruguay, Paraguay, might have consumed Alexis paraguensis, commonly known as yerba mate. And the notion here that I'm trying to get across is the fact that actually the use of psychoactive plants in modern society is very widespread. And certain plants, as the ones I just shared with you, we all take for granted have beneficial effects, or at least I do, I'm grateful for on any given day. Now, within those three plants that I just described to you, there are a number of small molecules, including this methylxanthine member caffeine that we recognize then as a stimulant of the nervous system. And we understand aspects of how that molecule works through engagement with a receptor called the adenosine receptor. And this is a class or member of a larger class of very interesting small molecules then, of alkaloids then. Now, the class of molecules that we're so excited about here in the context of the Center for Neuroscience of Psychedelics, of course, are a special class of psychoactive agents then, that as the name psychedelics tries to capture in so-called mind manifesting. There are molecules then with profound effects on perception, thought, and mood, and are showing incredible promise then clinically. These molecules, of course, have been used by humans for millennia, as long as there's recorded record. The special scientific challenge, though, as was introduced to you on the webinar last session, particularly from the work then of Bruce Rosen and colleagues using neuroimaging, is the notion that psychedelics can modulate aspects of neuroplasticity at a wide range of scales, not only changing, for example, overall connectivity patterns of neurons within the nervous system, but also aspects of their activity and down to the level of function of individual neurons. And as this beautiful review by Robin Carhart Harris and colleagues summarizes, the challenge for the field is to understand how is it that the engagement of a particular receptor by a psychedelic leads to that psychedelic experience then and potentially catalyzes therapeutic benefit. A specific example of this of great interest might be this small molecule, psilocybin, derived then initially from a fungus. And our concept here of using plants really extends then to our world of fungi and microbes as well for this. These molecules provide incredible tools then to gain insight into the mechanisms of brain function that, if harnessed correctly, we think have incredible therapeutic promise. What I want you all then to imagine then is the future. This isn't what exists right now, but it's the future we would like to work towards building. Let's consider then a hypothetical patient here, we'll call her Liz, that comes into the Mass General Hospital Department of Psychiatry to receive care. Liz might be suffering, as is common, from depression or anxiety or another psychiatric disorder. Working with the clinicians at MGH then to collect a family history and understand aspects then of Liz's unique case may lead then to desire to explore a psychedelic therapeutic. What we'd like to envision then is how the research that you heard about last webinar, namely the advanced neuroimaging, might provide insight then at the level of particular circuits that might be dysfunctional in Liz that might benefit then from a psychedelic therapy. Such research might also help us understand that individual for a diagnosis of what they might benefit from. But additional areas of translational research that we want to bring into the center then will involve the incorporation of genome sequencing methods to understand if there are particular genetic risk factors that might be predisposing someone to a psychiatric disease. But the technology that I'm most passionate about and excited to tell you more here that we think can have a transformative effect on our understanding of how psychedelics function and understanding their mechanism in a very deep level is one involving cellular reprogramming that lets us derive from a particular patient then through, for example, a skin biopsy, a sample that can be reprogrammed into a cell, a so-called induced pluripotent stem cell that can be differentiated into neurons. This remarkably then allows us to study an individual patient's pharmacology in the lab, in the dish, opening up this incredible opportunity to perform ex vivo tests of psychedelic agents. And in doing so, we hope this research then will be able to feed back and impact ultimately patient care. Now, the ability to create an induced pluripotent stem cell has existed for about a decade through the Nobel Prize-winning research of Yamanaka and colleagues and many others in the field that have really enabled this amazing ability to take a skin sample, or it could be a blood sample or another sample from a patient, and reprogramming it into this induced pluripotent stem cell. What's shown here is a colony of thousands of iPS cells growing on a feeder layer of cells. The cells like to be close to each other and interact with each other. But what's amazing about these cells is what we can do, and that is differentiate them into pretty much any cell type in the human body. For us, what's exciting then is this ability to grow human neural networks in the lab and do so in miniaturized formats that's amenable then to our efforts to screen and identify psychoactive molecules. What's depicted on this slide is one particular approach that we use in the lab, one that has facilitated our research by allowing us to develop these assays on a higher throughput scale. And remarkably, in a 2-week time period, we can go from these proliferating progenitor cells into these cells as depicted on the right that begin to form neural networks and form synapses as depicted with the individual arrows. We're able then to use robotic microscopy and high resolution and high content imaging methodology to extract quantitative information then about, for example, the length of the axons and dendrites and the number of synapses. This technology then enables us to test an individual patient's neurons then in their response to psychedelic agents. What's even more remarkable though, is we're not constrained to running these assays in a 2-dimensional world. Technology involving so-called organoid formation is letting us study mini brains or 3-dimensional organizations of these neurons that begin to mimic, although not fully recapitulate, the complexity of the human brain. If we were to take a high-resolution microscope and look at this little floating mini brain as is captured here in the Petri dish, we'd be able to detect then the different layers of cell types that are present and similar to those in the human cortex. This allows us then to study particular cell types that the neuroimaging and other approaches have told us are important for the response to psychedelic agents. In particular, the ability to create these so-called layer 5 specific excitatory cortical neurons then, a cell type that expresses particularly the 5-HT2A receptor, is one that's, we think, really exciting then for probing the activity of psychedelic agents. But more generally, we can study other aspects of neuroplasticity that have been implicated in response to psychedelics and may be critical for their long-term persistent effects. And that includes then activities at the level of neurogenesis, so the formation of new neurons, the alterations in axon dendrite structure that may contribute then to connectivity, and ultimately the formation and maintenance of synapses. These are the aspects of neuroplasticity that have never been able to be probed in a human context before, and the use then of stem cell and reprogramming technology allows us to perform these ex vivo scans. What we envision then is the application of these technologies, not just to Liz, but Liz is one member of a larger family or cohort of individuals that might be studied and enrolled in the clinical trials. We want to deeply characterize this patient cohort using the advanced neuroimaging methods that Bruce Rosen and colleagues spoke about, and their work then by our clinicians to understand individual cases. Our ability to derive from these patients a patient-specific stem cell then and characterize them deeply at the level of their genome, the transcriptome, the genes that might be turned on, their proteome, all of the proteins that might be expressed, and ultimately physiology will give us a deep understanding at an individual patient level. What we'd like to understand is the fact that there may be some responders in this population that respond to the psychedelic agent very well, but also some non-responders that don't show the desired therapeutic effect, or in some cases an adverse response. And we think that if we can derive then correlates in the lab that allow us to make these predictions, will improve ultimately patient care by helping our clinical colleagues choose the right drug for the right patient at the right time. Now, this is something we're really excited about as a technology and approach then for studying existing psychedelic agents. But we're also interested and excited to advance towards the development of what we consider next generation psychedelic agents. These would be agents that have an improved property and efficacy, the ability then ultimately to provide therapeutic benefit, and perhaps reduced side effects and increased tolerability. And the scientific question comes, where can we go about finding such molecules? And one approach, of course, is to take the existing ones and to use chemistry to derivatize them and develop new analogs. But as was introduced to you, we're inspired here to take an understanding or build on the understanding that comes from really the foundation of the psychedelic field. That is work by ethnobotanists such as Richard Evans Schultes, a very influential Harvard professor and educator who worked very closely with a number of indigenous peoples' populations in a variety of countries to really create an understanding from an ethnobotany perspective of many of the psychedelic agents that are currently of clinical interest. And Dr. Schultes then predicted here that the medicines of the future will actually come from our understanding then of the forest itself and use then of indigenous people's knowledge. Those of you that know Dr. Schultes know that he really inspired a generation of investigators to develop the field of ethnobotany. He himself is a legendary character that performed field work in the Amazon for over a decade, taking a leave of absence, if you will, from Harvard. In his travels then, he collected over 24,000 different plants, described more than 300 new species, and wrote over 500 papers. For those of you that want to learn more about Schultes' pioneering work, I'd reference you here to the Amazon team website here, which describes his journeys in great detail and some of the amazing findings of his research. Schultes' work then inspires us though through the principle of collaboration and inspiration then of interdisciplinary activity. It was this little book that Schultes, as a pre-med student at Harvard, first read, a book written by a psychiatrist written about mescaline, that inspired Schultes then to take the career path away from medical school, but into the field of ethnobotany. By the time Schultes graduated from Harvard with a master's degree and his PhD, he contributed then fundamental insights then into what we now recognize as the sacred cacti, such as peyote, Teonotocatl, the source of psilocybin, and Ololiqui, then the source of morning glories, really reflecting then three of the major classes of psychedelic agents currently of interest. And working with colleagues then, such as Dr. Albert Hoffman, Schultes was able to write these descriptions then that have now become definitive and inspirational guides towards the field of psychoactive plants. And in particular, this long-standing collaboration then between chemistry, biology, and psychiatrists have created the foundation that we're interested in building upon. When one takes a deeper look through the amazing writings of Schultes and others in the field, we see very elegant descriptions then of plants that at the time were very poorly understood, but of course now are of great interest medicinally. Schultes' own sort of inspiration, the botanist Richard Spruce, first described a series of plants, Banisteropsis caapi then, the source of the harmine class of alkaloids. Also a plant that's a member of that coffee family that we described at the beginning. Schultes then not only described these more commonly used psychedelic agents, but also in his writings a variety of less well studied but really exciting plants such as Tetrapteris methicica, for which his description of this is still true, that nothing is really known about it chemically, but has very similar effects to other types of ayahuasca that are more widespread and used. These then, and his writings, are inspirations for us to more deeply understand scientifically the nature of the psychoactive agents in each one of these plants. The fact is that we understand very little of many of these, and I've picked here 3 examples from Schultes' writings then that are of great interest to us. Ecorus americanus, or sweet flag, an agent that has medicinal and stimulant properties with possible psychedelic activities, is of interest because it's present here in North America, including in Massachusetts, although my trips on the weekends to local swamps and wet areas have not identified this plant, at least here in Boston. Another great example of a poorly understood but widespread genus of plants with potential psychoactive components here are Calea zacatini here, so-called dream herbs, that have been described then to have the ability to clarify senses, enable spirit communications, and lucid dreaming. Really a really exciting genus of plants then that requires further investigation. And beyond the more classical stimulants or aspects then of sort of psychedelic hallucinogens, this plant here, such as Scledium tortum, a plant then from South Africa that the work of Nigel Gerke and colleagues then have characterized as having potential mood enhancing, memory enhancing, and ability to combat fatigue, pain, and anxiety, is another example of a really exciting plant requiring further investigation. But what's most amazing about, I think, Schultes as an investigator and professor at Harvard is he continued to have a prolific effect on the field even after his retirement. And one of the last scientific papers that he wrote, 78 years old then, again, 7 years after his retirement from Harvard, summarizes the potential benefits for research then for neurodegenerative diseases such as Alzheimer's disease or Parkinson's and motor neuron diseases. And what Schultes, taking all of his research for over the 50 years throughout northern Amazon and other areas, summarized was of the 1,500 plants that are used for some medicinal purpose or as a potential poison, there were 25 different plants he suggested would be useful to study in the context of diseases of the elderly. One such example is this beautiful plant, Tabermontana herophilia, that is used by the indigenous people of Brazil then, that is consumed as a tea for roughly a 2-week time period, and is used then for individuals who are old and slow and forgetful. These are really exciting leads then to think about the potential psychoactive agents in these plants. What we want to do on the basis of this knowledge then is to really begin studying them, taking advantage of some of the advances in technology that just didn't exist at the time Schultes and others were doing their foundational work. So based on their writings then here, we want to systematically assess some of the plants that would really benefit from further study, creating a particular so-called neuronatural library. This will involve collaborations with traditional knowledge keepers, ethnobotanists, chemists, and psychiatrists and neurologists, carrying on that tradition of interdisciplinary collaboration that Schultes supported. We want to create then unique fractionations and extracts from each one of these plants, supplementing our library then with purified compounds and, of course, known psychedelic agents that are of interest based on ongoing clinical studies. We want to use that collection then in combination with those patient-derived stem cell models that I told you about to generate unique molecular and cellular signatures in our neurons and so-called mini brains. Again, for this work to go on, it's going to require close collaboration across multiple different areas of investigation, work with our psychiatry colleagues to point to the kinds of assays and molecules that would be most interesting to identify, work with our neuroimagers to be able to annotate the patient models as ones which might respond to a particular class of agents, and those interested in developing new medicines. What I want you to envision, though, is again the future here, taking advantage of some of the learnings that we already know in the psychedelic field, particularly an example here that comes from study of ayahuasca or yahe. The notion here that this medicine is composed really of multiple different plants, Banisteropsis caapi, that plant genus that Schultes and Spruce performed some of the initial characterization of, that's often taken in combination with another plant such as Psychotroia viridis, then a source of dimethyltryptamine. The combination of these two classes of molecules then represents this important notion of synergy, or in other words, an entourage effect through the combination of multiple molecules. What we'd like to do experimentally then is to really systematize this process of discovering molecules that can enhance each other's activity, a so-called systems neuropharmacology, which the systematic study then of the combination of these agents allows us to create a network where each molecule is represented by a node, and we connect them together with an edge that forms our overall understanding of psychedelic neural networks. Creating this map, creating this network then with the kinds of assays that we can now perform using patient-derived stem cell models, we think will provide a new foundation for the field of psychedelic medicine. To come back to where I started with here, to bring this again together, success in this activity is something that requires the unique type of interdisciplinary collaborative environment, the Center for Neuroscience of Psychedelics that we are building here at Mass General. That's because we need to work closely with our colleagues then in psychiatry to create models then from our patients that let us study their ex vivo response. We need to deeply annotate them with neuroimaging methods and genetic approaches. And we think if we can successfully pull these pieces of translational research together, we can ultimately create this field of precision psychedelic medicine. And I think this is a really exciting endeavor for a variety of individuals to contribute to. I want to end, though, with a quote, which is really a question that Richard Evans Schultes and Albert Hofmann posed at the beginning of the book that really influenced my interest in this area nearly 2 decades ago. And that was Schultes and Hofmann asked if we were able to perform a thorough understanding of the use of chemicals, understanding of the use and the chemical composition of psychedelics, would this not lead to the discovery of new pharmaceutical tools for psychiatric treatment or experimentation? And of course, we think the answer to this question is yes, and that carrying on the legacy here of Schultes, Hofmann, and others in the field of ethnobotany and ethnopharmacology positions us then to be able to have this impact ultimately on the treatment of patients. And with that, I'm happy to take some questions here from the audience. And I thank you again for spending a portion of your time this afternoon and going on this exciting journey with us. I'll just acknowledge then the fantastic colleagues and I get to work with here at Mass General Hospital, not only within my lab, but an extended network of collaborators then within Harvard Medical School and beyond. The inspiration for our research then comes from members of the community of ethnobotanists, ethnomycologists, ethnopharmacologists, and ultimately then traditional knowledge keepers and healers that inspire us to look in this direction. So with that, thank you very much for listening this afternoon.
Steve, that's really fun, and the promise of this research is really exceptional. A number of questions, and I hope we'll have time to get to them all. Again, for those who have more questions, use the Q&A function at the bottom of your screen. First question is that there's been hundreds of years of psychedelic use or psychoactive plant use. And some of those are now being studied as therapeutics and are widely used. How did they achieve ascendancy over the hundreds of other options, some of which you referred to? Was this chance? Was it natural selection? And I guess the implicit part of this question, is there a reason to believe we can do better, be more precise, or innovate by exploring all these other options that have not been used as widely?
Yeah, Jerry, that's an excellent question here of why are we so excited about a small number of plants right now? And I think arguably that's because of the focus on these and what's been demonstrated as their potential therapeutic promise. So when we think about so-called magic mushrooms or those species of psilocybe that produces psilocybin and related agents in it, I think one of the reasons those have been more widespread has been, again, the perhaps ease of culturing and growing mushrooms culturally across the area. These are agents then that didn't require synthetic chemistry in the early days to produce. Although, of course, now understanding what the psychoactive principle is, synthetic chemistry can be used to produce those agents in a purified and standardized way. The same is true, I think, when we look at components then of ayahuasca, such as the dimethyltryptamine, which again is being explored then potential therapeutic applications. So there's this interplay, I think, between understanding the plant, its use, chemistry to elucidate the active principles. And in some ways, the current research then is focused on these examples then of the major classes that Schultes first described and of aspects of phenylethylamines, the ergot alkaloid class, and the tryptamines. And so the question is, are there others out there that perhaps have benefit, that have been overlooked? And if you read closely some of Schultes' writings, he would say, yes, there are a number of plants that have just for either historical reasons been less well characterized because they may be localized in a unique geography, a unique culture, but have the potential then to have just as much benefit then when applied in a therapeutic context. And so I think that's the excitement in the field, is the examples we have right now are a roadmap, but can we improve on them? Can we make them more well tolerated? Can we reduce the side effect profiles in some cases? And can we make a greater percentage of the population respond to them? And I think the answer to that is yes.
Can you extend that roadmap a little bit on the journey beyond your lab and how what you're doing can eventually become therapeutics? Can you say more about what has to happen next after you curate this incredible library of opportunities?
Yeah, so I think one of the exciting aspects of using a plant that has already been used by humans is that we understand something about the safety and toxicology of them. Not everything, but in many cases, we're able to learn that a plant, for example, isn't going to produce an addictive substance or produce one which is necessarily ultimately toxic to the body. And so that's a real step forward in the therapeutic discovery process. But it requires then close collaboration and interaction then with a chemist, for example, that might be interested in synthesizing a subset or a series of molecules from that, and exploring then ultimately what we learn about the targets of those molecules. And so an example might be that most of our psychedelic agents that we are of current clinical interest are targeting the 5-HT2A receptor. So imagine that we uncover and discover a different receptor, a different target then, that perhaps has some unique advantages therapeutically. I think there are already clues when we look at plants like Scledium and others that of course are not targeting the 5-HT2A receptor, but have these other effects then that we wouldn't characterize as traditionally hallucinogenic, but are clearly important for impacting neuroplasticity. I think it's kind of expanding the definition of psychedelic to incorporate new mechanisms and new activities that's important there. And that may lead to the recognition that there's an existing natural product that can be utilized, but it might be that identification of a target then that allows us to engage in a more traditional drug discovery process then, as the pharmaceutical industry would, to optimize synthetic compounds for them. I'm a little agnostic about both, and I think approaches, and I think though the inspiration of a natural product though keeps us focused on exciting molecules.
Now, I was going to ask you a question about what your favorite plant is, but you just referred to it, and I think I know the answer, but maybe others would be interested to know.
Yeah, I'm really excited by this plant, Scledium torsum, that Nigel Gerke from South Africa has characterized in great detail and written about, with its potential effects then for aspects of mood, well-being, anxiety, and overall energy. And this is an interesting example because right now it's not a scheduled substance. It's not something that is controlled then in the same way that a psilocybin or DMT or other agents are, and is really targeting, I think, a really critical area of investigation of thinking about agents then to help with disorders such as treatment-resistant depression and anxiety. And it's just following the descriptions then of this plant and thinking about some of the mechanisms. And there are a few alkaloids that are known within this plant that may be mediating these activities. And those targets and mechanisms then are really exciting to try to understand. Can we develop on the basis of that assays using our human neurons that report on their activity? And again, this is interesting where a plant, of course, is already consumed and used by people, but we just don't understand the neurobiology and neuroscience of it yet. By going backwards, then we can begin to fill in those pictures. But I have a long list of plants that, again, are really exciting about. And if you were to ever open...
Make you pick a favorite. Here's a very,
like,
very important question. I'll read it. It says, how does the center intend to create a reciprocally beneficial relationship with indigenous groups to whom we are indebted for carrying the knowledge of these psychoactive plants over the centuries? Critical question as the work may result in patenting and profiting off related molecules.
Yeah, that's an excellent question. In this area, we have to be mindful of the importance of following some of the conventions, such as the United Nations Convention on Biodiversity, that requires us to consider the kind of equivalent notion of an informed consent and our ability then to consider access to the resources that are created from such a product and investigation. And I think the answer to that question is to really work closely with those in the area which are traditional knowledge keepers and elevate their voices through understanding it is their knowledge that is inspiring us and working towards us, inspiring us in the directions that we want to go. And I think there are ways and examples then of how to do that, to think about it proactively, to provide a sharing of any possible resources that are created, whether they be financial or access to the medicine then that may not be present then. I think it's an important one that also, a question that also touches on aspects of conservation, recognizing that some of these plants, particularly within the Amazon, are right now very, very much under stress from destruction of the environment. And one of the, I think, greatest legacies of Schultes and colleagues then was really to point towards the early value of protecting these areas and protecting this knowledge then of indigenous cultures and people. So the short answer is we'd love to work with people that want to help us do that correctly and effectively then towards new areas.
There have been a number of questions that have touched on something you referred to in passing, which is the possibility of the knowledge derived from exploring these the novel molecules derived from these plants for non-psychiatric conditions. There was a question about, can you imagine a therapeutic that might help people with coma or neurodegenerative diseases or Alzheimer's? How might you can imagine that outcome?
Yeah, so, again, one of the exciting aspects about Schultes' writings in particular were the description then of these 25 plants then that he suggested would be of great value for studying in the context of what we would consider to be an Alzheimer's disease type dementia. And remarkably, since that publication, as far as I can tell, there's been very little investigation of many of those plants. But if we actually take a step back, the use of psychoactive plants has already played an important role in development of treatments for Alzheimer's disease. And the reason we have acetylcholinesterase inhibitors such as galantamine comes from our knowledge then of particular plants that have the ability to inhibit acetylcholinesterases. And so that's one mechanism. That's not a disease-modifying mechanism for treatment of dementia, but is an agent that can provide benefit to patients. And so using the kinds of patient-derived stem cell models that we've already generated in the lab from patients with a variety of neurodegenerative diseases, so tauopathies and dementia, we can create models then that really model aspect of the disease and use those to screen for new agents. And I'm amazed going through and reading, of those 1,500 plants that Schultes and colleagues described, the diversity of potential applications beyond maybe infectious diseases and areas, but skeletal muscular disorders, paralysis, a whole range of agents. And again, as they are often referred to, very little characterization to none has been done on the chemical components. And so if we believe, I think, in the successes of discovery of agents like psilocybin, we should believe then in the potential then for moving forward with characterization of these agents.
One quick question was, can you repeat the name of that plant that we referred to as your favorite? And that was the Scaledium tortuosa, right? Is that?
Yeah, I mean, I'd be saying
we don't really want to say it's your favorite because you love all your children equally.
I love many of them because I'm fascinated by them. I'm fascinated by the potential and the history of each one of them. And I'll give you another example of one that I think is really interesting. And that is, we all understand that ayahuasca is composed of multiple plants, an admixture then to the initial Banisteropsis caapi, the source then of the monoamine oxidase inhibitor. But there are also descriptions of other agents added, for example, to facilitate after the psychedelic experience a memory of that activity. So perhaps what we would put in maybe psychiatry terms, helping with the integration of that therapeutic event, as if someone is able to access that memory later then in their life. And such a plant then has already been described in the Amazon as being added to ayahuasca mixtures. And so I think as we understand what is it that would actually have the most benefit for patients, is it that kind of agent that has the ability then to prolong the experience and potential therapeutic benefit for a patient? Or perhaps it's the opposite. Perhaps it's a fast-acting agent that has really an impact for only maybe minutes that facilitates the process then of access to a broader set of patients in a clinical setting. And so I think, again, answering those questions requires this close collaboration with our clinical colleagues to define. But again, I'm really amazed each time I sit down each evening and look through some of the writings in the field, not just of plants, but also our fungal friends then. I think there's some amazing starting points for discovery here.
Your last answer sort of begs another question, which is, medicine for many diseases, whether it's cancer or hypertension or even psychopharmacology, we often, to get the best result, have to combine agents that have similar or sometimes overlapping and sometimes different mechanisms. And in nature, many plants, often there are multiple compounds of it in the plant that's been used naturally, and the so-called entourage effect and so forth. So will your work, involve the complexity of looking at some of these possibilities in combinatorially? I mean,
I think that's what's exciting about being able to grow these neural networks in tiny wells, right? And by tiny, I mean might take 100 microliters of volume, but we can manufacture hundreds of these little neural networks or hundreds of these 3D little mini brains. And it's that miniaturization and the ability to extract out information from them that lets us play that game, for example, of looking at all possible pairwise combinations of agents. And if you go through the math and think about maybe 100 plants and you were to look at those in combinations, that's over close to 5,000 different unique combinations then to look at. And as the size of that library grows, if we fractionate it or identify unique components, it is in that search space, and I'm really excited. And that's this notion of building that network. rooted under the psychedelics that we understand right now, but looking then for something that can enhance the effect of psilocybin or enhance the effect of that scaledium agent then. And my gut feeling, and this remains to be seen, but inspired by other work that we've done in the lab, that doing that then can really provide something very beneficial in the long run for this development of new therapeutics. And that's what we mean by next generation psychedelics. And if we've done so using humans, patients then, as our framework for that information, I think it's got a greater chance of impacting patient care.
I want to also tell our participants that if you've sent in a question and we don't get to it, we will respond to it after time with an email. But there are some interesting questions about the idea that that we're kind of medicalizing the knowledge from these plants, and presumably that would mean that these compounds would be developed through traditional pharmacological testing and approval and so forth. And I guess the theme of some of these questions is that shouldn't people just be able to access, compounds that exist in nature without having to go through the medicalization of them? So, I mean, I realize that's a complicated issue, but maybe you can take it on.
Yeah, maybe I think there's sort of two parts to that question that I think are interesting to think about the benefits, though, of so-called medicalization, of these. One might be to address the point of access to certain agents that may not be present throughout the world in all sort of contexts then. And that's, I think, harder to keep in mind when sitting in such an amazing area such as Boston and the rich biomedical resources that we have around us. But I think thinking through how to provide access to agents becomes an important benefit then of such an activity. And the second, and I think this is one of the most important, when I think of my own use of medicinal plants and herbs and nutraceuticals then, is to be able to have a better sense of what it is that you're actually consuming then from it. I'll give you examples then of trying to access for research purposes then components of scaledium that can be purchased off of the web. And unfortunately, in many cases, what you're able to access really isn't the pure plant or pure agent. And so when trying to develop medicines that are going to be safe and ultimately effective to patients, there are some benefits, I think, of what we're able to do then in standardizing and understanding what it is that you're consuming. And I think that has to be balanced, though, with this notion of appropriately using the information and recognizing the sources of information that we have that come from ultimately indigenous people's knowledge. And so that's a dialogue and something we're excited to take on with other leaders in this area that have thought about this, how to do it effectively. And I think there are some great examples to follow.
Okay. There was a question about questions about how you get your questions answered through email. If you ask your question anonymously, then maybe follow up with a email directly to Molly McCarthy, and she'll be able to have your question answered. Here's a question about a plant called the Syrian rue as a far more accessible and widely available source of harmine and harmaline. Do you have any comments on that?
Yeah, Syrian rue is a plant that is currently accessible from a variety of different sources. In fact, was how we first discovered chemically that harmine molecule that was later recognized to be identical to the compound that was called telepethine, present in Banisteropsis caapi. And it's a great example where multiple different plants produce the same psychoactive component. Then this is a monoamine oxidase inhibitor, which can potentiate the effects then of, for example, dimethyltryptamine. I think it's a great example, though, of a plant that may have other components besides the harmine within it that are actually, to date, not that well understood. And so when a molecule like that is made, the plant is biosynthesizing it. It's a genetically encoded small molecule, if you will, and may make a variety of other agents that I'd argue today we still don't understand all of them. So even a plant like that, which in some sense has been studied and used in society for a long time, I think there's new things to learn from it. And that would be a great example of one.
So could the work you're doing be relevant to people who are on psychiatric medications? There's a lot of concern, even when you mentioned scaledium, the products that contain it will caution about use with psychiatric medications. Will the work address the potential interaction with drugs that patients with psychiatric disorders may be on?
Great point. You know, when I think about mental health care, it's something best done working with experienced and talented clinicians and physicians that have the experience and knowledge of how to guide such activity. So that is because in some cases, then we don't understand the interaction of these natural products with maybe existing medications. And so discussing that then with a psychiatrist then would be an important part of that care. I think though we'd like to think of agents that, for example, maybe can potentiate the activities of an existing therapeutic. And so in part of my life, I've spent a long time thinking about lithium as an example, used to treat bipolar disorder, but a molecule, or really not a molecule, an ion here, which we don't understand its mechanisms. But could we find another plant which potentiates its activity, allowing you then to perhaps lower the dose of lithium that you'd be giving to a patient then, and doing so then reduce the potential toxicity that's seen or restore sensitivity to it. And so I think if we can conceptualize the challenges of what it is that would have a greater impact on patient care, the example I think that's so exciting of the potential value of something like psilocybin in treatment-resistant depression is, by definition, it means then often a patient has already not responded to a variety of classical pharmaceuticals. And so having impact in those challenging and intractable contexts then with a potential single dose or limited number of doses combined with psychotherapy, to me, that's the dream of a new kind of psychiatry then that changes the model or interact of how we provide patient care. And I think that's what's really exciting to those in the field then, is this perhaps overcomes some of the limitations of our traditional psychotherapeutic options.
Steve, you're an extraordinary teacher, and this has been a really informative program. One thing I do want to mention, and one question raised this issue, I want to point out that Steve's work in the chemical neurobiology lab is one part of the center. And there are other parts that have been the subject of our webinars, the neuroimaging, the clinical neuroscience. And for example, an important compound that we are studying in other contexts, like MDMA, is not part of our ethnobotany presentation, but we are obviously committed to and interested in the whole field of psychedelics, and have proposed studies and associated neurobiology and neuroimaging around MDMA research as well. And we don't mean to say that we're only focused on plant-derived potential therapeutics, but really on the whole space of psychedelic-derived therapeutics. So I want to make that clear, because for those of you joining for the first time may think that we're just about plants. We are very committed to the area that Steve, and this is only part of Steve's work as well. This is not all that Steve does for the Center for Neuroscience. And so I know we're coming to the end, and I want to just first of all thank Steve again. He's an extraordinary colleague and scientist, and the field of psychedelic neuroscience is privileged to have him working on its behalf. The early work of our center, before the neuroscience of psychedelics can compel traditional funding like federal grant support, will principally be accomplished through private support, including philanthropy. And I want to thank our current donors for partnering with us early on, and want others to know that support for activities like Steve just presented will require substantial additional investments from others who are committed to this vision. And if you want to learn more about getting involved in our work, feel free to contact Dick Simon or me directly, and we'll be happy to talk about opportunities in this area and how to participate with us. But especially thank you very much for attending. Again, a recording of this webinar will be available later in this week, and please share it with your friends or any who may have missed the live presentation. And with that, I want to again express my appreciation to all who have come together to make this center a reality, to make it a powerful instrument for the future of this field, for all of those who have been interested in supporting, and for the team behind us that have made today's program possible. So have a wonderful rest of the week, and we look forward to seeing you in November, when our next webinar will feature Dr. Sharman Ghaznavi, who is a cognitive neuroscientist and a psychiatric clinician, who will talk about other studies that we're doing, for example, on treatment-resistant depression, rumination, cognitive neuroscience, featuring psilocybin and other compounds. And I hope you will join us then. There'll be a save the date notification coming out to all of you on our email list. So thank you all again for attending, and see you again.